to our third speaker, Tom Blundell from the University of Cambridge, again from United Kingdom. And he will talk about the structural biology, bioinformatics, and drug discovery from carrier to bacterial infections and COVID-19. We are looking forward to his amazing talk. Tom, the floor is yours.
Oh, thank you very much. I'll just share the screen. Is that working? I need to go to
yes, we can see it. We can see it. That's good. If you do it full screen, we'll be good to go, Tom.
Yeah, I've clicked it for full screen. I'm just waiting for it to come back again.
No worries.
Good. Can you see it now? Yes.
If you can, we see green grass so far. Only green grass, with some, I think, rain on it.
I can't hear or understand what you're saying.
Basically, we don't see slides. We only see green colored grass.
Green colored grass.
Is that your background? Yes, might be your background, Tom.
The background is no background.
Do you mind going back to your PowerPoint?
My PowerPoint is. Sorry, what do you want me to do with it?
If you go to your PowerPoint, click on the PowerPoint, and at the bottom,
I'm in the show then. I had the screen full up.
Yeah, I think it might be working now. Just a second. If you can click, yes. So now go to, yes, where the structural biology, bioinformatics, drug discovery is written. Yeah, click on, yeah, exactly. I think you're clicking the right location, slideshow. Yes, you're right. Click on it again, yeah. Tom.
Well, that's what I did before. That's what I did before. Let's see whether it works this time.
Maybe continue as if you're presenting, and let's see if it's moving forward. If you can click a couple slides, we will see if it is moving or not.
Yes. It's gosh, I don't know what's happened now.
Okay. Let's go back to that screen if you don't mind, Tom. I think we will figure it out. No worries. If you can go back to that screen.
I can't get back again now.
Okay.
Oh yes, okay, I'm back now. It was for me okay earlier on. It's now got completely.
If you can share the slides on the chat box, maybe that might be a way. If you can do a share.
I did do that.
Okay.
Safe again.
If you can share the slides on the chat box, then I can share the screen for you. Sometimes I can help that way as well.
It just seems to have gone completely now.
No worries. Take your time. Take your time. No worries. We will figure it out. So one way is to either yourself going back, turning all the other background things. Maybe you have too many background stuff, We can start with closing the other things. Sometimes that confuses.
I removed all that.
Excellent job. And then
if my screen's gone completely dead now.
That's okay. You will be able to open it. Don't worry.
I can't get it open at all.
Okay. No worries. We have time. No rush. Take your time. We are here to support you, Tom. No worries.
Okay.
You can even turn off and on the computer. We will wait for you, if that is gonna help.
No, that's gonna be helpful.
Yeah, but the best way is, as I mentioned, if you have challenges in opening your PowerPoint, you can share the slides on the chat box. Here, there is a chat box where we're all talking. And if you share over there, share the screen for you as well.
It's all right for me.
There you go. You got it. You got it. Perfect. Perfect. Okay. We're on. Tom, the floor is yours. Excellent.
Well, I'm in my sixth decade of working and learning, so sorry about that.
No, you're doing
Uh,
amazing. Thank you so much. Thank you, Tom.
Okay, so apologies for all of that. And so what I'm going to talk about today is really two kinds of tools that have impacted on us very recently in drug discovery. I'm going to, however, give a historical background. I'm afraid when you're approaching 80, then you can't escape history. So the two tools that you'll see are themes in my talk in drug discovery, which have really come in very strongly over the past years, although we've been using them for quite a long time. Is one is cryo-EM in the imaging, and the other is AI. And in fact, we've used the first software I wrote using machine learning methods was actually quite a few years ago. We started focusing very strongly 8 years ago. The cryo-EM has been going for many, many years, but both of them have been very fundamental advances or seen fundamental advances that I'll illustrate today. So what I'm going to do is to talk, of course, about human genomes, and in that respect, cancer, as well as off-site drug targeting. I'll talk obviously about some of the targets in mycobacteria. I'll focus on leprosy, tuberculosis, and Mycobacterium abscessus, which infects cystic fibrosis patients. And I'll then go on to talk, as I said, a little bit about the resolution revolution in cryo-EM, which is allowing us to do drug discovery. So both in my company called Aztecs, we took half the building down and rebuilt it to use cryo-EM. And in my academic lab, where I'm most of the time, we have the whole of the basement full of cryo-EM machines. So here's a result from the last few months about a multi-component system involved in DNA repair seen through cryo-EM. And this is the 6 different components seen in molecular detail at high resolution. So these begin to provide information about targets. And this cryo-EM resolution, you'll see, you can even see a tiny fragment of molecular weight 300 in a huge protein with in one of the cases, over 4,000 amino acids. I'll then go on just to mention what we've done over the last year on the SARS topic, where we have used the methods we developed for many other areas. I'll talk a little bit about finding drug candidates and what we've done in my company called Aztecs. I shouldn't say it's my company. I co-founded it with 2 colleagues, Haran Jyoti and Chris Abel. And I'll mention just how AI has begun to predict affinities of ligands and impact on almost everything we're doing. So despite having been in the area for a long time, we're up to date in using a lot of machine learning in this area. I'll very briefly look at emerging resistance, where we've used machine learning to understand the impacts of resistance mutations. And we've also used, well, that's in fact the machine learning methodology summarized. So that's an overview of what I'm going to say. I just go back historically and say that the most influential time in my life in thinking about drug discovery and development and lead optimization was due to this lady, Dorothy Hodgkin. I can't resist always showing a picture of her because she started doing what I picked up on insulin. These are insulin crystals, which he first started working on in 1934. I joined 30 years later in 1964. And this is the X-ray diffraction pattern. I don't use X-rays much at all anymore because of the cryo-EM resolution revolution. But this was what really influenced me very much. It was our structure of insulin, which we published eventually in 1969. So you can see, what is it, 50 years ago. Here's a picture of Dorothy Hodgkin and in discussion. And I usually show this one, as I often comment, to show how beautiful I was nearly 60 years ago. So that's where I started. And Dorothy Hodgkin, although she was very much left-wing, as indeed I was a left-wing councillor running the city of Oxford in parallel to doing this work on insulin, she was also very much connected to industry and introduced us to all sorts of companies to obtain insulin and other crystals. After insulin, I guess the main point that I want to just emphasize is the HIV protease became, I guess, the first example where I could really see the pressure on in going through drug discovery. We had already ideas about interpreting the HIV polyprotein and then identifying target proteins in there. And of course, the main target was the HIV protease. Other people worked on it. I predicted and published in Nature 10 years earlier than this paper that it would exist somewhere. And it was good to find it in HIV and use it as a target. New drugs came very quickly, but of course also did resistance. So that's the sort of background of which influenced me very much. All through that period, I was working with Pfizer, Zeneca, before that ICI, and other companies, as well as being in academia. So I just want to go through a little bit now the paradigm for structure-guided drug discovery to discuss how it's been influenced by these recent developments. So I always look at it as two steps. One is exploring biological space. How can we find out about the possible targets in the whole genome? And I'll talk about the computational methods we've developed over the years to go from genes to proteins and get 70 or 80% of the gene products modeled, and at least allow that to evaluate their drugability. And then, of course, the second step is screening chemical libraries. And I guess the major step that we've taken forward was the idea of fragment-based discovery, where you have a much smaller library of very small fragments, which we developed in our company starting in 1999. And now with 2 drugs on the market, one selling $500 million a year. So I'll talk about those 2 aspects. But let me first talk about biological space. And I just focus very quickly on one system, which is non-homologous end joining. This is a typical kind of target because it involves multiple steps: synapsis, end processing, and ligation, and altogether 10 or more different gene products, and 3 steps: synapsis, end processing, and ligation. So this is DNA repair. And I just wanted to give you this example because over the years, over 20 years or more, we've used various methods, small angle X-ray scattering, molecular modeling, X-ray crystallography, and then cryo-EM to define the components of this system, and of course things like mass spectroscopy. But the real revolution that, as I said, I wanted to emphasize has been in cryo-EM. So to get that X-ray structure, I worked for over 10 years, publishing a low resolution structure in 2010, and then a medium resolution structure, which is the one you see in the middle, at 4.3 angstrom. That took me another 7 years. And then in just a year or two using cryo-EM, we have even higher resolution models. That was 4.3 angstrom. And using the cryo-EM resolution revolution, we now have models at 2.8 angstrom resolution that we could get not in 10 years, not in a year, really very, very quickly. And this is the very large protein called DNA PK. It's, I think, the largest protein kinase with 4,000 amino acids. And this is the structure with cryo-EM. We published it in Nature and Science earlier on using X-rays. And this is what it looks like with cryo-EM, not at 4.3 angstrom, which we got with X-rays, but with cryo-EM 2.8 angstrom. And this has really been a complete resolution revolution. And almost all, as I said, the drug discovery we're doing now is based on cryo-EM images. And this is just to show you how you can get things that I couldn't possibly get with X-ray. And this is the work of Amanda Chaplin, Steve Hardwick, and Tonya Kafala. And here you see a 6-component system in DNA repair that we've been able to capture using cryo-EM. And we've taken this to bits and we've looked at the druggability of the different components. So that's the first point that I want to make, that you can make these incredible developments using cryo-EM now. You don't target just single units, you can target multi-component systems. And what most biochemists have ignored for many years is that almost everything that happens in the cell of interest has more than 10 components in it, very complex systems. So, of course, that's experimental. And what we've done over the years is to write a lot of software as well. You can see that some of this was written over 30 years ago, Composer. I guess Modeler, which now has, I think, 11,400 citations, is a piece of software we've used for homology modeling, which I first wrote about in Nature in the mid-80s with Janet Thornton and Mike Sternberg and Lynn Savanda. And Modeler is very useful to get an idea of druggability for the rest of the genome, but I wouldn't use it as a reliable test for or target for drug discovery. It tells you about druggability, but not reliable enough. So cryo-EM will do that, but you can look at the druggability and you can develop models using Modeler, and then Fugue uses methods to align sequences and structures to get the best information for homology recognition. And so we've devised a whole series of software, which we then use to develop the knowledge of the proteome for drug discovery. So, for example, tuberculosis, which we worked on for quite a long time, has around 4,000 gene products. And we use our automated software to develop models which are quite reasonable for around 65 to 70% of the targets for all the gene products. and then you can assess druggability and decide which ones you're going to study using cryo-EM or X-ray. We've done this for tuberculosis. We've done it for leprosy, which is an area I've got into work over the last 3 or 4 years. I was giving a major lecture in Colorado with several thousand people around me, and I finished my talk standing on a pedestal. And then at the end, after the questions, I looked down and saw this group of people looking at me intently. And I thought, gosh, Tom, you really shouldn't talk about these things so confidently. What have I done wrong? And of course, it wasn't that, it was the leprosy mission arriving around the pedestal to ask me to work on leprosy. So that's another area very much ignored by the West because it's a stigma, and most countries won't admit the number of patients who are in these leprosy colonies for all their lives. And so that's the second area of target. And we've also looked at cystic fibrosis targets, but recently we've used the same modeling techniques to look at the SARS-CoV-2 3D models. And this is just a quick review of what we've done over the last year. We've modeled all the proteins and their multi-component systems. Many people have just published protomers, but you need higher oligomers. And then we've been using this for not only, as I said, the oligomeric modeling, but binding predictions, mutation analysis, and docking hypotheses. I know a lot of people have done this for SARS-CoV-2, but we were able to do it in 2 or 3 months after we found out that it was going to be a major challenge in the last year. And so this is the kind of techniques we've used, and the analysis. We have an interactive database where you can go in. It's completely accessible to look at all the components, oligomeric structures, and so on. So that's the COVID-2 area. And of course, there again, cryo-EM has completely changed things. So without my really being aware of what's happening in one corner of my lab, I find 2 of my colleagues, Shereen Thomas and Shikang Liang, have got a very high resolution structure of one of the cryo-EM, using cryo-EM of one of the SARS-CoV-2 proteins. And this is better than we've been able to get with X-rays in our lab anyway. And we can use that for drug discovery. So the other thing that I want to emphasize is the AI. And so in another part of my lab, we've developed AI, and I'll talk a little bit about it briefly later, for looking at mutational analysis, which we've been doing for 8 or 9 years. But this is using AI deep learning to predict protein-protein interaction sites. We've also used it in looking and assessing protein affinities for ligands. And there again, the point that was made earlier in the talks is that the major challenge with machine learning is not writing the software. You can do that in a day. It's actually making sure that your learning set is reliable and extensive. And so we spent most of the last year with our AI trying to extend the data sets and learning, and making sure that if we're assessing using AI affinities of ligands and predicting them, then we've learned on reliable sets. So I'm not going to go through this in detail. Many people have done similar things, but it's obviously a very exciting area. So that's defining the biological space. Just very briefly defining the chemical space. Then of course, from the 60s, 70s, and 80s, I developed with other people structure-guided drug discovery. But in the 90s, with this guy, Haran Jyoti, and also Chris Abel, who was a chemistry collaborator and sadly died a few months ago, but very much driven by Harron, we developed the method of fragment-based discovery. The idea here, as you know, other people had developed it slightly before us, but what we did is to use it in a structure-guided sense. And in 1999, founded a company, Aztecs. So instead of having a library of multi-millions, we have a library of less than 1,000 fragments, molecular weight 300, where you can explore the chemical space. I started off with 3 people in the lab and 2 above the local shopping center to get this project going. And we very soon expanded to 80 employees and built the building you can see there on the Science Park. And then pretty quickly after that, by 2012, we had a whole lot of drugs in clinical trials and running out of money, and we had to sell it. So I was rather surprised to find that we could sell it for nearly $1 billion from just this idea of small fragments. And so the company was sold, but the interesting thing is that they kept everybody employed. It was bought by Otsuka, a Japanese company, and they kept me as chairing the science advisory board. So we've taken this fragment-based method. Our first milestone was 5 years ago with a drug for advanced breast cancer, which then got approval in 2017, jointly with Novartis. What we had to do in a small company is we passed the drugs as we designed them on for the clinical trials, usually after phase 1. And so phase 2 and phase 3 are usually funded by larger companies. And then we had another drug. We've got actually 2 on the market. This is for metastatic urethral carcinoma, but the one we have, ribosectin, is actually selling $500 million a year. So quite an interesting development. So I just wanted to say that what we've done in academia is, of course, to fill in where companies can't operate because there's not enough returns. So tuberculosis, even with huge numbers of people dying, because they're dying in developing countries, they're not very attractive to large pharma. The leprosy is an acute case because nobody is even aware of the problems because of the stigma, as I mentioned before. Cystic fibrosis is another problem which really has to be addressed partly in academia, simply because there are, in a genetic disease, very few people have it, so not many companies have been confident to go into it. Although in cystic fibrosis, there have been advances in classical pharmaceutical. So we've looked at targets in cancer, we've looked at targets in neglected disease, and I've got special pleasure out of working on this because it's an area where there's been great neglect. And about 15 years ago, we had a visitor, and that was Bill Gates. He came to my company and asked us to work on tuberculosis. Of course, we couldn't do that in the company because the people who need the drugs can't usually pay for them. So we spun an image of the company back into academia, into my lab, and started working with Bill's support. And this is particularly important to me because in this picture, in the right, you'll see a beautiful Zimbabwe in Africa. On the left, you see this lady in the middle. That is Dr. Sabanda. She's my wife and partner. And here are my 2 daughters. And they've been very lucky being in, the daughter's lucky being in the West, in the UK. But you see down here, my grandnephews are still in Zimbabwe, and we've had major problems in Zimbabwe. I've lost relatives through tuberculosis and, of course, HIV as well. It's been a major challenge there. So just very quickly, we've done all the things you do in drug discovery. We've got a whole range of first-line and second-line drugs already available, but we've used our biophysics-driven fragment-based discovery with, again, a multinational team, gender balance, by the way, and developing using biophysical screening methods. And Bill Gates visiting, keeping in touch, coming to see us. I made him put a lab coat on, and here he's challenging some of my colleagues on what they were doing in the lab, still with his lab coat on, you can see. And out of these studies, we've managed to get lead compounds for tuberculosis. The real problem is getting anybody to move them through clinical trials or to get them at phase 2 level to be used without going through phase 3, which is very expensive. So that was tuberculosis. We worked on Mycobacterium abscessus. Here's a beautiful slide of a drug that candidate that we've just developing in Mycobacterium abscessus. So that will be for cystic fibrosis. And here is again the revolution from cryo-EM. I never believed that we'd be able to see a fragment using cryo-EM, but you can see here you get a beautiful heme in this molecule. You can see the fragment of a fragment size molecule. We've been now over the last few months with a very strong program developing kinase inhibitors using drug discovery at cryo-EM. And we've done this in academia and in our company. Just I'm probably way over time, but I just want to mention that the other thing we've been doing using machine learning is developing an understanding of mutations. And this, I first started using machine learning 8 years ago for understanding the emergence and the impacts of mutations, giving drug resistance, so on. And what we've done is to develop two kinds of programs. One is a statistical methodology that I published 24 years ago, I think, something like that, originally 1997. And this looks at a statistical analysis of the impacts of the mutations on the folded and unfolded state. This software has been very widely used over the last 7, 8 years in particular, but it was actually first written in 1997. And then, of course, we've used a whole, we've developed a whole suite of programs using machine learning, AI methods, and this is our one. It's using graph learning data. I've still found that that's insufficient really to look at all the features of understanding the impacts of mutations on ligand binding, for example. So that's the story. We've got examples of resistance that we've understood, and we're thinking about how we can now use that information and the mutability of other parts of the molecule, which might be less to design drugs that will be less impacted by resistance. And so I leave you, I've probably gone over time, but here is my group. You can see that it's certainly it's multidisciplinary, but you can see that it's reasonably gender balanced. But what I'm very pleased about is that it comes from all over the world. In fact, when it was slightly larger, I discovered at a party that we could do 35 languages in my group. We're so, we're collected from all over the world. Of course, Indians, for example, have many languages, so it's not all different countries. But of course, I have a lot to thank my wife for, and she's from Zimbabwe, as I said. And I think that's the lesson I learned from Dorothy Hodgkin, that science must be international, interdisciplinary, and gender balanced. But with all of that, it can be fantastically exciting. So I leave you with that overview. Thank you very much.
Thank you so much, Tom, for this amazing presentation. I really appreciate it. We are open for any questions at this stage. The floor is wide open for Tom. Tom, this was really impressive, you know, what you have presented. I really appreciate it. Any questions for Tom, Dr. Goldblum, or anyone who wants to chime in for Tom's presentation?
I have a question.
Please go ahead, Shahar.
Yes.
Hey, first, thank you for the last slide. It is a, I appreciate it when I'm not, you know, when people not just to do, but also say why they're doing the things that they're doing. So I really appreciate that kind of message. But my question is a bit different. I would like to understand the difference that you see between X-ray and cryo-EM. How similar are the structures and how different are the structures? And I know that each group or each technology shine at different groups of proteins, but when you do the same, when you use two methods on the same protein, how similar are the structures and what do you learn from both?
Yes, well, that's something that's concerned me over the last, I think, 20 years. Well, actually, we got our first cryo-EM machines multiple decades ago when I was in London. But looking at those very high resolution structures that I described, then for the DNA PK, we get a pretty good alignment of the structure interpreted from the cryo-EM and the X-ray. What is different is when you start impacting on it with mutations and the compatibility with crystals becomes more difficult. And so, for example, in the picture I showed you of CAT-G, one of the enzymes involved in tuberculosis, there the mutations actually cause local realignments and restructuring. I can see that in cryo-EM, but if I do that, I can't get the crystals to form. So what I found is that cryo-EM is particularly good for understanding systems where either through binding small ligands or having mutations in, you can actually get conformational change. So that's where we've seen the gain. But I have to say that we're still doing fragment-based discovery, although we've started trying it with cryo-EM, but that's the only area where we're really still focusing on X-rays. Just about all the other structural work now, even with relatively small molecules, proteins, you know, 30,000 or something like that, I thought weren't going to be good targets. But even with those, we can see a lot of information using cryo-EM. So I'm afraid the resolution revolution of cryo-EM has moved me on. And so I must say it's been a very exciting few years. having all my assumptions on computational biology and on experimental structural biology upturned, and having to accept that there are other methods which are really effective.
Thanks. Thank you very much.
Thank you so much. Any other questions before we move on? I think if there are any questions for Tom, please go ahead. This is the best time to ask the question. If not, Tom, I want to thank you again. We learned amazing things from you. Thank you so much. We're really grateful. Thanks a lot.
Well, thanks for inviting me, and sorry about the technical problems at the beginning.
No worries. You did phenomenal.